thank everybody for logging in today, and I want to thank Ruching and Tom for inviting me. And this is actually following Philip. This is a very low-tech approach, and a lot of the basic technique came in through histology in terms of the decalcification of the bone that's necessary. So just to give you a very brief history of me, I'm the Senior Director of Microscopy and Image Analysis at Weill Cornell Medical College in New York City. We are far from our mothership in Ithaca. And I've been here since 1988, when it was just an EM facility, and over the 3.5 decades, it has grown to include a wide array of optical microscopy technology as well. But this is EM, and this is what I will present. So we're all used to fixing, processing, embedding things like muscle and liver and kidney, and we know that we have to preserve it, infiltrate it with a resin to make it hard enough so that we can section it thinly so that we can image it in the EM. And then we're presented with bone, which starts off hard and difficult to infiltrate. So it presents a major problem, as we talked previous questions asked about damaging your knife blade, and then how do you cut it all? And so that's due to the hydroxyapatite matrix. And so we have to use methods to remove that. And again, a lot of this was also used in basic histology technology, but we've adapted it to work in EM. And the method I'm going to present, I got from Dr. Steve Doty when he was at the Hospital for Special Surgery. Steve was a consultant for NASA about what happens to astronauts' bones when they're in space for a long time. And he is a very valuable resource. He has retired, and I miss him greatly. So the protocol that Steve shared with me uses a chelating agent, EDTA, to remove the mineral. There are other methods that are available using acids and other things to dissolve the matrix, but those are much harsher. And the EDTA is very gentle. And when you use it on tissue that has been well fixed, it doesn't do anything to the ultrastructure of the other tissues, the surrounding tissues, or the bone matrix, the bone itself. Oops, sorry. So if you're working with small animals, you can do your initial fixation by whole body perfusion, as shown in this diagram. And that's, you can do a femoral artery or a left ventricular puncture and flush. It's a good idea to flush the system first with ringers so that you don't get blood clots in the capillaries, and that allows the fixative to perfuse well through. And then once you've done the whole body perfusion, you can isolate the bone that you're interested in and get it into the decalcification. So there are commercial systems that you can purchase, and these are developed mostly, I guess you can see these have histology cassettes in it that use the chelating solution, and they're somewhat automated, but we're a little old school here, and so just putting the tissues in conical tubes with EDTA solution that gets changed every other day. And depending on the size of the bone, I'm sure that cochlea would not have taken the full 2 weeks that the femurs that I have worked with did. But as long as you keep it agitating or rotating in this case and change the EDTA, you get a very nice decalcification. And when the bone is fully decalcified, it's rubbery. So here is a picture of I believe this was a mouse femur. Those are not my hands. I got this off the web. But it shows you that the bone actually does get very rubbery. You could, if you wanted to, tie it in a knot. And, as long as you've gotten all the hydroxyapatite out, it'll infiltrate well and section. And if you miss a little bit, you'll still see the hydroxyapatite in the bone, and your knife will suffer a little bit. So maybe you want to use an older knife. So I'm just going to go through the protocol. Sorry for the text-heavy slides, but since Ruching is going to share these presentations with you, I thought it would be helpful for you guys to have the protocol. So we use a primary fix that's a modified Karnofsky. So it's a 2.5% glute, 4% PFA, 0.2% picric acid, which makes our life safety people here very nervous. And we perfuse and then overnight fix the isolated bone, buffer wash, and then we use the decalcification buffer. And after things have been fully decalcified, we will use a secondary fix of osmium. And this is the full protocol, stepwise. So again, fix overnight, isolate the bone, put it in the EDTA solution, change that every other day, make sure it's agitated until you get your rubbery bone. And certainly if you're unsure, you can stop the rotator, take the bone out, see if it's bendable, put it back if it's not. And then one of the lovely things about this, because as a core, we get samples from researchers throughout the medical school and from other neighboring institutions, and sometimes they can't always get their samples to us in a timely fashion. With the decalcification, you can hold that bone in the EDTA solution almost indefinitely with no detriment to the sample. Then once you receive it, we cut the samples down to the area of interest, and we process pretty much normally, just with extended dehydration steps and careful stepwise infiltration into the resin, embed and polymerize. This protocol still uses propylene oxide. In my lab, we have switched to acetonitrile. It does not become explosive, and it's just a safer chemical in general. And then here's a low mag of a section of bone. And all of these bone pictures, I want to thank Dr. Matthew Greenblatt from the Pathology and Laboratory Medicine Department here at Weill Cornell. We've done a number of projects with Matt, and he very generously allowed me to share his images with you. So here's as I said, a low mag map through a femur, and you can see blood vessels, you can see the matrix, and I'll go in a little higher. Here again is that blood vessel, the capillary, and you can see even with 2 weeks in the EDTA solution, the ultrastructure of the cells is still very, very nice. And you can see the matrix. Here we have those few hydroxyapatite crystals that were still remaining in the bone. And then all of the surrounding connective tissue. And here again, going higher mag into the cells to show you, here's rough ER. You can see all of the nice ribosomes, nucleus, overall the structure. Here's some collagen. So the nucleus is the cells are very well preserved. And it was a short talk. I thank you all. Happy to answer any questions. And again, I want to thank Dr. Doty for providing the original protocol. My EM specialist in the lab is Juan Jimenez, and he handles, I would say, 95% of the EM samples that come in. And again, to Matt Greenblatt and Joan Sun for providing us with the images. And if anyone has questions that they think of after the fact, here's my contact information and the website for my core. And that was me. So, any questions?
All right. Thank you, Lee.
I know it was short, but made the point. Thank you.
We've got a question for you.
Okay.
Go ahead, Mark.
Thank you, Lee. Maybe there's one thing I could add that might be useful for some people. Many years ago, we did a study where we added glutaraldehyde to our decalcification EDTA solution. The goal was that we're studying the extracellular matrix and the mineral crystals, the hydroxyapatite in the extracellular matrix. And as we dissolved the mineral in EDTA, we wanted to make sure that we kept any mineral binding proteins or extracellular matrix proteins where the original fixation didn't penetrate. And we did a quantitative immunogold study just to show that, in fact, you have better retention of extracellular matrix proteins if you add some aldehyde to your decalcification solution. So if you're doing immunolabeling studies, you have to be careful how much you add, but I think it's a very good thing for those in the field to add a little bit of fixative to the decal solution. It was just a comment. Thank you.
Oh, thank you. That's very helpful. And I have a new project with Dr. Greenblatt, and actually we will be doing some immuno. So I'll see. I didn't have the gluten in my EDTA, but we'll see what happens. That's very helpful. Thank you.
Oh, hello. I'm sorry. I'm kind of new into the whole electron microscopy sort of thing. I've been doing an apprenticeship for a year and all that. So this is more of a question just out of like interest. So normally I work with soft biological, like tissue samples and all that. When you said about you use EPON resin, I'm just wondering, out of pure curiosity, is it the do you use BDMA1, MNA, DDSA, and EPON 812, or is it a different kind of epoxy sort of
No,
combination?
we use an EPON analog. So we buy most of our EM chemicals from Electron Microscopy Sciences. So our EPON resin is Embed 812. And then, yeah, DDSA, NMA, and BDMA as the accelerator.
Oh, perfect. Okay,
And
thank you very much.
we do use the hard formulation for it, so that it even though the bone has been decalcified and is rubbery, we do want to try to match the hardness of the resin to the tissue. So we do a hard formulation.
Yes, that makes sense. Thank you very much.